Hi, my name is Catharina, and today I present my talk entitled "HNF4-alpha deletion effect on beta cells is influenced by sex and age." So briefly, I want to talk about the role of HNF4-alpha in beta cells. It was originally described in MODY1, that is a type of diabetes mellitus that originates in the mutation of this gene. This mutation is associated with beta cell dysfunction, that is, the inability of beta cells to adequately produce and secrete insulin. Beta cells are within the pancreatic islet that is responsible for partially regulating the secretion. As of today, we know that HNF4-alpha affects beta cell proliferation, differentiation, as well as its ability to secrete insulin. However, we still need to better comprehend why and how this influence happens. And to do that, we need to take into account two key factors for diabetes mellitus establishment: age and sex. Therefore, the aim of our work is to evaluate the influence of sex and age on beta cell dysfunction induced by HNF4-alpha knockout. To do that, we used Krellox mice that were mice that had a beta cell specific HNF4-alpha knockout that was induced during puberty. In these mice, we collected the pancreas for immunostaining assays. At 21 days of age, the mice were weaned, and at 40 days of age, we conducted the first glucose tolerance test, after which the knockout was induced using tamoxifen. At 50 days of age, we conducted the second glucose tolerance test, and at 90 days of age, the third one. At 50 and 90 days of age, we collected the pancreas for immunostaining. So for our results, we have glucose tolerance tests in which at 40 days of age, we saw no difference in glucose tolerance between the knockout and the control mice. At 50 days of age, we saw that knockout mice had a reduced glucose tolerance when compared to the control mice. Lastly, at 90 days of age, we saw that the knockout mice had glucose tolerance reduced when compared to the control mice, and additionally, the male mice had a more significant reduction when compared to the female mice, suggesting that sex plays a role in the glucose tolerance impairment. When we stained for HNF4-alpha, we saw, as can be seen in this image, that there was a reduced positive stain in the knockout mice when compared to the control mice, as highlighted by the arrows. And when we quantified this stain, we saw that there were reduced positive nuclei per islet in the knockout groups at both ages. When we stained for insulin, we saw that the positive stain was localized in the center of the islet in the control mice, as we would expect, and it was reduced in the knockout mice. When we quantified the stain, we saw a reduced percentage of positive insulin per islet in the knockout mice at 50 days of age, and at 90 days of age, this reduction was more significant in the male mice, and we had an influence of sex in this reduction. When we analyzed the beta cell mass, we saw that there was no difference at 50 days of age, but at 90 days of age, we had a reduction in this parameter for the knockout mice and a tendency for the influence of sex, suggesting that insulin reduction is a parameter influenced by sex in our knockout mice. When stained for glucagon, we saw that the stain was localized in the border of the islet in the control mice, where we would expect to find it, and in the knockout mice, it seems to be less peripheral. When we quantified the alpha cell mass, we saw that it was elevated in the knockout mice when compared to their controls at both ages, and the male mice had a significant increase when compared to the female knockout mice. So for our conclusions, age is an important factor influencing both beta cells and islets' response to HNF4-alpha knockout. Additionally, sex influences hyperglycemia progress as well as loss of insulin-positive area within the islets. Therefore, we conclude that both age and sex collectively influence the response to the HNF4-alpha knockout. Lastly, I want to thank everyone from my lab as well as their funding agencies for their support.